I welcome you all to my class. Today we shall be talking on a very interesting topic, and that is how the development of embryo takes place from a zygote in case of dicot plants. We have seen how zygote comes into existence, isn't it? Bring to mind embryo sac and then the pollen tube, and how fertilization and double fertilization takes place, and zygote and triploid endosperm comes into existence. Now, from zygote we shall exceed forward and see how it develops into an embryo. Now, just think like how do we make sprouts? You soak the gram seeds overnight, and then in the morning we cover it in a piece of cloth, and say next morning we get nice sprouts, isn't it? Now, this is the process of embryo development. Now here what happens, here the seed the zygote is dormant. Now when it gets a favorable condition, that means water, then it starts developing. Now what happens? So this is zygote inside an embryo sac, and that is again in the seed ovule. So zygote, when it gets good condition, it starts division. Now the first division, mitotic division, now henceforth all the division will be mitotic division. First mitotic division gives rise to two unequal cells. Upper one is basal cell and the lower one is apical cell. Opposite. Basal cell, apical cell, they are in books sometimes referred to as CBCA. Now basal cell does not take part in embryo development. It gives rise to suspensor cell. Suspensor cell, the function of suspensor cell is to push the developing embryo more and more towards the center, so that it is well protected and well nourished. Basal cell gives rise to a suspensor, and apical cell gives rise to embryo. So this is the first division. Then division proceeds. The basal cell will divide transversely and give rise to a row of cells, whereas apical cell will divide longitudinally and will give rise to two cells. This is a two-cell stage. Then suspensor divides like this. The top cell of suspensor is known as hausteria, which is the biggest of all cells, and the last, which is in contact with the developing embryo, that is known as hypophysis. Now hypophysis has got a function. It develops into root cap. Hypophysis develops into root cap, for short notes. So now we have four-cell stage. Four-cell stage is known as quadrant. Now we shall be concentrating only on the embryo development. So the quadrant, again it divides one more division and gives rise to 8-cell stage, which is known as octet. Now octet stage has got a speciality. Here we get the root and stem cells. Root and stem cells are demarcated in the octet stage of embryo development. From octet stage, mitotic division is continuously going. We get lot of cells, and here in next stage is globular stage. It becomes ball-like, and outer layer is demarcated here. There is a distinct outer layer, and the further cells are inside that outer layer. Outer layer is known as dermatogen, and it gives rise to epidermis. Whereas all other parts of the embryo will come from the inner cells. Now, as the development proceeds from globular stage, we get heart-shaped stage, and here the two protruding part, they will develop into cotyledons. Heart stage will give rise to horseshoe, when the cotyledons will be more prominent, which I haven't shown here. Cotyledons will become more prominent. Then finally we will have a fully developed embryo, and here we will have two cotyledons. Then we will have a plumule and a radicle. Now if this is kept for division is seen further, then from here we see the root and shoot coming out, the one which we see in sprouts. So, and this is the suspensor, hausteria, hypophysis, and then the fully grown embryo. So this is how embryo development in case of dicotyledonous plant take place. Here the specimen which is used for study is Capsula bursa pastoris, which is a plant which belongs to Brassicaceae family. It's an ideal plant to study the embryo development in case of dicot plants. So this is how development of embryo takes place in dicot plants. Let's revise. Zygote, from zygote we get two-cell stage in which the cells are unequal in size. basal cell and apical cell. Basal cell will give rise to suspensor, apical cell will give rise to embryo. Then we have transverse division and longitudinal division. Basal cell goes transverse division and apical cell undergoes longitudinal division. We have 4 cell stage which is known as quadrant, then 8 cell stage which is known as octant. In octant stage we get root and shoot cells. Then we have globular cell stage where we get 2 layers, outer layer is dermatogen and all the other layers are inside that dermatogen. Then we get heart shape stage, from there we get horseshoe stage, and from there we get fully developed embryo with radicle and plumule and the 2 cotyledons. And also the suspensor cell. This whole thing is inside the ovule, which is the seed. So this is all, my dear students, for today. This much we have learned how an embryo develops in case of dicot plant.